and go. Alright, so here we have our double helix model of DNA. And if you look over here, this will explain what we have, our basic setup. One line is orange and red gummy bears. The 5' end is the end with the red. 3' end is the one with the orange. On the other side, 5' is green, 3' is yellow. Since they run anti-parallel, you can see that yellow and red, yellow being the 3', red being the 5', are on the same side, just as orange and green are on the same side when they're 3' and 5'. If you look back to the key, adenine is a purine model, so it's double in the DNA. It pairs with thymine, which is going to be the orange. That is a single, guanine, double, and then cytosine is a single. Now, for DNA replication, what happens is the helicase comes through, and as it comes through, it's going to split up these models right here. The nucleotide bases are being split, and they come off to the side. At the same time as the helicase is coming through and splitting everything up, moving it to its side, you have what are called binding proteins, single-stranded binding proteins. They come through to stabilize the outside sugar and phosphate, and they will end up back here. Now, since we have this end, and it's 5' out here, and this end with the 3' out here, the 3' end is going to be the leading strand. Since you build 5' to 3', this one can continuously build this way. So we're just going to add in our nucleotides here, which is done by DNA polymerase 1, or 3, sorry. My bad. DNA polymerase 3 is going to come through, and they're going to add from right to left for me. I'm not sure what direction it is for you. DNA polymerase through comes in, we'll add this, and at the same time as it's adding in the nucleotides, we're going to be able to add in the sugar and phosphate molecules on the end. And likewise, you've got 5' end here, 3' here, 3' here, and 5' here. That makes this new strand of DNA the leading strand, anti-parallel. The other end, since this is 5' and you have to build 5' to 3', you're going to have to go from segments. DNA polymerase 3 is going to come in, start with an RNA primer. Start right here, and as it comes through, it's going to pair one little segment right here. And then it's just going to keep following back, going right here. This is called the Okazaki fragments, sorry. DNA polymerase 3 keeps going through, keeps adding an RNA primer so that it can build. And eventually, we'll get right here. Now what happens as you get here is you get DNA polymerase 1, which goes through and corrects. DNA polymerase 1 going through will correct any on the leading strand fairly easy. DNA polymerase 1 going through on the lagging strand, as it goes through, is going to take out the RNA primers. It's going to also help add in the sugar and the phosphate and replace any of the RNA primer with DNA. Just keep these separated for a little bit. DNA ligase is the final enzyme that goes through the lagging strand, and what that does is it just fully forms the strand of sugars and phosphates so that it can connect, and then the Okazaki fragments are connected, and you now have 2 full strands of DNA.